So I don't think I need to remind this audience of the importance of antibiotic-resistant infection or of sepsis, and I don't think I need to remind this audience that we're now increasingly looking for alternative therapies. So amongst the alternative therapies, the one that is often at the top of the list is phage therapy, which we'll spend most of our time talking about today. So like bacterial antibiotic resistance testing, phage susceptibility testing can be done in liquid medium or on broth medium, and essentially we're looking for inhibition of bacterial growth because we know that that correlates with clinical outcomes. So in many ways it looks similar in the diagnostic laboratory, but in so many ways it is different. It's different because this is a living therapy. We're talking about, as many of you know well, I think, a viral infection of bacteria. Most of the viruses on Earth are parasitic of bacteria rather than humans. And so they enter a life cycle which looks much like the one we're familiar with viruses that infect humans, with all the different stages of their life cycle a target for resistance by bacteria, but the most important of which is the initial attachment and absorption of the phage. And this is often the target of our diagnostic testing. So we know that the mature bacterial virus or bacteriophage enters, much like we know viruses enter any cell by initial attachment and penetration. And the most mature interaction is probably one of integration into the host chromosome, with switching to a destructive cycle, the so-called lytic cycle, when the bacterial population is under stress. So that makes sense. And we also know that the integration of the bacteriophage brings with it extra capacities for the bacteria, the most famous of which are the toxins in diphtheria, in Staph aureus, in cholera, the changing of the surface serotype of Shigella, and indeed the introduction of antibiotic resistance genes into the chromosome occasionally. But what we're looking for when we're seeking to use a phage for therapeutic purposes is to seek out a phage that is exclusively parasitic. And this is probably the less adapted form, if you like, because ultimately a completely parasitic virus will destroy its own food source and therefore its own chance for survival. So many bacterial viruses have adapted to try not to kill the host completely, but we seek a purely parasitic virus that destroys the host and destroys it quickly so that we can use this for therapeutic purposes. We know also that if we add therapeutic viruses to antibiotics, we get an effect that is greater than the sum of its parts, that it is true synergy. So it appears that the bacterial response to an antibiotic or a virus so injures the bacteria that the secondary attack of a virus or an antibiotic that's added to it gives a greater effect. And so this synergy is a great advantage of using antibacterial viruses or bacteriophages, simply because most of the time we will also be using an antibiotic, often an antibiotic that doesn't work as well as we would like, and that's why we're seeking a bacteriophage. Clearly, to use bacteriophages as a therapeutic option, we need to embrace the advantage but also the great challenge of using bacteriophages, because it can reasonably be said that for every bacteria there is a virus. So over the 300 million years or so that bacteriophages and bacteria have co-evolved since before mammals evolved, then they have diversified. And so that very diversity means that for a therapeutic option, we need a lot of different phages. So that is a major challenge that needs to be met by biobanking, but can, to some extent, be met by careful choice of the phages, because we know that, in fact, the diversity amongst the successful bacterial pathogens of humans is actually relatively more limited. So that we know, for example, there are only, half a dozen or less bacteria that dominate lethal infection in humans, and inside each species there is maybe only dozens of different subtypes that are probably responsible for 80 or 90% of the infections caused by that species in humans. So an intelligent biobanking program can make good choices about the sort of phages that you can use. The other big issue, of course, because this is a living therapy, is its viability on the shelf in the pharmacy. And so one of the great challenges in bacteriophage therapy is to create a bacteriophage product that is still fully viable 6 months or a year later. So in our country of Australia, we've set out to try and address the first problem by assembling diverse collections of phages. Now, most of us, I think, experience bacteriophages in the research environment. Certainly, that's where my experience of them is. And so the use of them as therapies is constantly being drawn from diverse collections around the world, largely for research labs. But to assemble all these, we need a common phage description. And that phage description, from a clinical point of view, needs to include a description of the bacterial targets that phage will destroy, so the phage host range. And that needs to be a standardised description, so that, let's say, for example, the golden Staph aureus, we need to understand the subtypes of Staph aureus that this particular phage will destroy. And obviously we would choose phages that would destroy the most common of the subtypes of any given species as our therapeutic first options. And so the choice of a phage host range is very important when you're setting up your biobanking structures, and we believe that you should standardise the bacterial hosts that you test your phages against for each bacterial species, and ideally use a surveillance program to inform what those bacterial host sets should be. Similarly, obviously, you need to describe the phage itself carefully, and the most important thing is to be confident that it is exclusively lytic. That is to say, it is not likely to integrate into the host, but rather simply to destroy it. And that kind of information is readily derived from the DNA sequence of the phage. So simple genomic sequencing solves a lot of those problems. The other important point, given that synergy is so important in bacteriophage and antibiotic combined therapy, is to understand the synergistic effects of the phages of choice against the bacterial targets using the antibiotics that are normally used for those bacteria. So if we're talking about Staphylococcus aureus, for example, there's a small handful of antibiotics that we would commonly use for that. And so we want to know whether the phages that we would put up as our preferred therapeutic phages are synergistic with those antibiotics against those important organisms. The next important issue is obviously purification, because one of the key issues here is that, unlike, say, a vaccine which you grow in a mammalian host, we are growing in a bacterial host. And so if you are unable to purify that product well, you risk introducing bacterial components, including very inflammatory components such as cell wall components, lipopolysaccharide, into your therapeutic product. And clearly you don't want to give someone a phage preparation that contains bacterial cell walls that might induce a strong inflammatory response. So the purification process is important. At the moment, many of us would use a range of physicochemical processes that may range from simple density gradients, such as a cesium chloride gradient, right through to more conventional filtration, anion exchange, chromatography type techniques. And I think the scaling up of this towards the classic good manufacturing process probably requires the latter techniques. So if you look at the companies that are now trying to produce therapeutic phages, they're using classic GMP style processes. The actual provision of the phage to the patient, we think logically, or I think everybody would agree logically, needs to follow the routine path. A diagnostic test is provided on the basis of a bacterial isolate that generates a prescription by a suitably authorised prescriber, and the pharmacy fills the prescription, dispenses this prescription. The pharmacy needs to understand phage safety and production strain safety, so they need to be able to verify the product as being safe for use in humans. And we think that the phage itself and its safety, the presence of resistant genes and so forth, and the production strain that it is made in need to be regulated and certified. So clearly, if you want to make a cholera phage and you make it in a virulent cholera strain, because that's the only strain it will grow in, and you put cholera toxin in your preparation, the pharmacy and the patient won't be very happy about that. And so you need a mechanism to monitor and license that risk. Similarly, obviously, there's the manufacturing safety itself. So did you produce a phage that was uncontaminated, that was high titer, that was exactly what you said it was, and doesn't contain any bacterial byproducts such as lipopolysaccharide. So with these in mind, what we've done is try and get a group of phage researchers and clinician scientists together, so a group of doctors and scientists together who believe that we can see a way forward to make this available, because we are, like everyone in the world, I think, concerned about the lack of new antimicrobial options available. And to do that, we originally started with a group of us who were already working with phages, mostly on a research basis, but starting to treat humans. And we reached out to colleagues around the world who were doing the same sort of thing to try and make sure that everybody was, I guess, thinking the same way and developing the same approach to this, because one of our great concerns is not to repeat the mistakes of the past with antibiotics by having unregulated and uncontrolled prescribing. So our mission is to establish trials to understand the best use cases of phages and to understand their cost effectiveness, and to try and establish the best practice by providing high quality phage therapy, high quality materials, that is clean, reliable phage products, by providing prescribing protocols so that we were all using it the same way, and by monitoring what they're doing in the same way. So we asked the Australian infectious diseases prescribers by survey what they thought was most important in terms of clinical use. So that's the top green and blue section A of this slide, and what they thought was most important in terms of the kind of pathogens or infections we should be treating. So when we asked them, they said, look, 90% of the respondents said that they would be happy to use therapeutic phages, that they have people that they would want to use it on now. More than 90% said that they would use it inside a clinical trials framework. And most people said that they really wanted a clinical trials framework to provide some discipline to the use of phage therapy, which in Australia at the moment is completely unregulated. So it's regarded as an experimental drug, and any prescriber, if they can obtain high-quality phage, can approach their hospital for permission to use it as an emergency. So our prescribers around Australia said they wanted to see safety data, they wanted to see efficacy data, but they wanted clean protocols about how to use things. And the target organisms are the ones that everyone around the world thinks. It's biofilm infections in prosthetic joints and devices, and it's very resistant infections such as the gram-negative, such as Mycobacterium abscessus. So I guess there were no surprises. And the way we went about trying to solve this was to establish a protocol which was directed towards process. So what we said was, given that the phages may be a GMP product from a company, or they may be a cesium chloride preparation from a colleague from overseas, the product itself is not what we want to regulate. The drug committee of your hospital will make sure that product is safe to give to the patient. And our therapeutic regulator will give us permission. But what we lacked was a protocol. So we set out a routine protocol, which everybody agreed on, that requires rigorous gatekeeping. So you have to see an infectious diseases specialist before you can enter the trial, and they must verify that all current therapy is optimised. And then you have to undergo a standardised monitoring protocol under informed consent, and all the data is directly reported to the regulator. So this gives us a mechanism whereby any physician can use rescue therapy and still sit inside a clinical framework. We published a standardised monitoring protocol that includes common sense things like looking for resistance development in the bacteria, doing the kinetics of the phage therapy on a regular basis so we understand the dosing strategy. And our first publication suggested that the kind of doses that we, and in fact most people use, are probably about right. We also monitor the human immune response. Now, this is rather expensive to do, of course, so you wouldn't necessarily do it on every patient, but it gave us some confidence that we were seeing the right sort of therapy occurring. So in this slide, we see a single patient. Their human transcriptome is represented in panel A, and you can see that there are some genes that are thought to be related to the adaptive immune response, and some that are related to the innate immune response that are differentially regulated over time, so that we see quite a distinction between the pre-phage on the left-hand side of the top panel and the later days. So we can see here as this upregulation fails until we eventually get to the point, and you can see in the screen below this clear trajectory of an adaptive immune response, which is takes a while to develop. We saw antibodies, in other words, developing typically around the 10-day mark, which is what we'd expect. And we saw the innate immune response initially upregulated in response to LPS release, we think. So it's clearly a TLR4 pathway type of phenomenon. So if you compare this with our other discovery, with the other antimicrobials that have been discovered over time, I think we would argue that the beauty of bacteriophage therapy is that it covers really, as far as we can tell, probably all bacteria. Which really not a toxic product if you prepare it cleanly. And its very high specificity has been its Achilles heel, because until the modern age of diagnostics, we really didn't have capacity to handle something that was so specific. Which is fine if you have a specific organism that you want to deal with. But what happens when you have gene sharing? Because we know that, in fact, most of antimicrobial resistance is in fact acquired by gene acquisition, so plasmids. And we know that these are shared and transmitted, so that rather than a phage targeting a whole subtype of bacteria, we have bacteria sharing plasmids. So the flip side of this coin is we need an approach to plasmids, and most of the antimicrobial resistance that we're worried about is on the big plasmids. So these are big plasmids that are the same genome size as a phage, and they encode their own transmission between bacteria, and they determine their own epidemiology by a set of rules that govern which bacteria they like to live in, which plasmids they are prepared to live with, and these plasmids are the ones that are spreading all of our carbophenamases. They carry the KPC, they carry all the things that we're most worried about in the enterobacteria, which dominate the WHO hit list of antibiotic resistance threats. But because these bacteria have rules about the way they live in a bacteria and the way they are transmitted between bacteria, and the fact that they tend to exclude each other to protect their own ecological niche in bacteria, these systems can be subverted. And there's plenty of evidence that we can do this very effectively to extinguish a bacterial plasmid from a population of diverse bacteria that all share that plasmid, simply by designing another plasmid that uses the same rules and that can outcompete and displace that plasmid. So I've given some examples there below on the bottom right of publications that describe this, but essentially what we're talking about is using a bacterial plasmid to displace an unwanted bacterial plasmid by very specifically targeting it. So it's the flip side of the phage therapy coin, where you're targeting a specific bacteria. This time you're targeting a specific resistance problem. And you do this by exploiting the mechanisms that are used by those big resistance plasmids to manage their own stability. They have a system which, if it's removed and they are having to rely on simple cell division, will result in their loss from the population, their segregation out, because simple partitioning of the bacterial plasmids with the chromosomes is only about 60% to 70% efficient in most plasmids. And so without these special systems, addiction systems if you like, they're lost. So you can use these systems against them. So in both of these instances, and this is why I talk about these two together, in both of these instances, you're talking about the host ranges of the particular, whether it be a plasmid or a phage, and these relate to largely to receptor specificities on the cell surface. And these things, because they have grown up in nature with bacteria since before humans evolved, have their own exclusion and incompatibility rules, which we can exploit and subvert. And we now have the tools, the modern diagnostic tools, to use that. They may deliver genetic elements, and certainly we know that phages are important in delivering virulence elements such as cholera, such as diphtheria. We know that the plasmids are important in delivering resistance elements such as all our most important common emerging carbophenamases. And we know that both of these things have design systems to make sure that they don't get segregated. So we have to deliberately manipulate them. If they're plasmids, we can deliver them in a bacterium by simply swallowing them. If they're phages, we can deliver them as a preparation, and often we're using them intravenously, but we can deliver them by any route. And if we're talking about plasmids, just again, we're talking about cleaning a population of bacteria or cleaning an ecological niche by using the rules that determine that niche occupation by a plasmid. But for a phage, we're talking about very specific one bacteria at a time. So sometimes we just want to get rid of a Pseudomonas from a person. Other times we want to get rid of a KPC on a plasmid that's spread through 3 different bacterial species. So where are we at the moment with these things? I think everybody's aware that the phage story on the right side of the screen has been shown to be safe in humans, it's been shown to be safe in children, and it's been shown to be safe in very, very sick people. And there are publications that I've pointed to that are quite recent that give us, I think, good confidence and that I've already referred to. And this is a 100-year-old story, so there's a lot of anecdotes of safe use. This is now moving into clinical trials. The reason I think we're not seeing a lot of it is because of the commercialisation questions, which is probably a topic for another day. On the left-hand side of the screen, the flip side of this coin is the plasmids. Now, this is not done in many places. There are some publications that I point you to there, and we're still early days. This is really probably first in human, just coming up now. So this is the future, if you like. Phages are now, plasmids are the future. How can we reach out at the moment? I think as a global community, we would say that the only thing we can offer from Australia at the moment is that we would reach out to our colleagues who are interested in joining us, and we would at the moment exchanging phages between each other. We're happy to sequence people's phages if they want them. Obviously, they retain ownership of those phages and those data. We would welcome collaborations in clinical trials. I've got a picture of Randir Paz and Ronan Hazan from Israel on the bottom, just to exemplify this, because they've got a terrific phage that was used for the first time in a child in one of our hospitals, and we all published together, and that's how I think we do things in a global community. I think it's earlier days with the plasmid story, but we're very interested in collaborations there, and so I think I should stop and ask if there's any questions now, and thank all those people that you can see on the screen that I've mentioned in the bottom left-hand corner with photographs who've done all the work. So I thank you for your attention and look forward to dealing with any questions.